plants. Talking about beetles today. I'll be talking to you about sexual selection for social network position among multiple populations. Before I get started, I would like to dedicate this talk to my PhD advisor who passed away a few days ago, Elena Tuttle. She was really influential on my work as a scientist and my development as a human being. Conspecific interactions are among the most important and common events in an individual's lifetime. Sexual selection and behavioral ecology have made great inroads in understanding the evolution of these behaviors, but largely by focusing on dyadic interactions. But many of us realize that these behaviors don't happen in a dyadic vacuum. They actually happen in a large population where individuals are interacting with each other throughout the course of their lives. And one of the tools that we can kind of start to begin to grapple with this complexity is a tool called social network analysis. And so if you want to look at social networks, this is a picture that we can use to represent them visually. And so each one of these dots is an individual, and the thickness of these lines represents the number of times they've interacted together with each other. One of the reasons I think network position, so an individual's location in this broader network, is interesting is because it's the expression of that phenotype, of that network position, is a result of the characteristics of the focal individual who you're looking at, but also the behavioral expression of the social partners inside that larger network. Then the individual network position, so where an individual lies in this network, can be mathematically described, and it's a description of how individuals interact with their entire social network. And so we can kind of simultaneously look at all these interactions in one analysis. Network position, so your location in a network, has been shown to predict how individuals receive information, how information flows through an individual, or also how diseases are transmitted and the role that individual plays in that transmission. Indeed, a number of studies have shown that an individual's network position co-varies with their fitness. This has largely been done with a mating success component of fitness, but some of them have also shown this for survivorship. Almost all of these studies have looked at this, though, in the selection for network position in a single population over a single breeding season. So what we thought we would do is look to see how consistent is selection for social network position among populations, so across space within a single breeding season. And then kind of a rough cut, what we're trying to do here is to see if there are factors that predict the magnitude and direction of selection among these populations, if they do indeed differ. So there are lots of different ways that you can describe an individual's position in a network. There are hundreds of them, actually. The two that I will talk about today are both measures of what we call network centrality, so how well individuals are connected within their own network. And so the first one is called strength. You can think of this as a weighted number of partners. So if we take this focal individual, this one has a high strength because they've got lots of connections and they've had repeated interactions with those social partners. The next one is called betweenness. This is also a measure of centrality to a network, but really you have to have the entire network mapped in order to measure this. And so this is, if you take any two individuals in the network and draw the shortest path between those two individuals, you do that for everybody and you see how many of those paths pass through your focal individual. And that's a measure of its betweenness. So how many paths pass through that particular node that you're interested in. Our study system is the forked fungus beetle. See, not an ant. They're kind of a classical system for strong sexual and social selection for body size. The males have these horns that they use to combat with other males for access to females, and that's under strong positive selection. They remain largely on a single subpopulation for an entire breeding season. So if I go out in the woods, this patch of fungus on the dead tree is kind of our subpopulation. And they have important social interactions, such as male-male combat and an extended and complex courtship between the males and the females. The majority of these behaviors happen on the surface of these brackets and on the surface of the log. So I can go out with a small army of undergraduates and we can survey an entire population over the course of a day and see all of the social interactions. One of the other nice things about this system is they have a really stereotypic mate guarding behavior, where the males climb onto the back of the females and they face head to head. And this has been shown to be a very reliable indicator of insemination success. So this is going to be our fitness component to define mating success. We've done this before in a single population, so looking at copulation success, this mate guarding, and looking at some of these other network metrics, we've shown that they co-vary with fitness. But again, this is just in a single population. We wanted to look, is this true over multiple populations? So we surveyed 9 populations in a single meta population, a single old growth forest. We went out twice a day and we did scan sampling for 64 days. All individuals in all these populations were captured and labeled and photographed for body size measurements. And during a scan sample, we would find all the individuals we could, and their location and all of their social partners were recorded. We define social interactions as coming within a few body lengths of each other. We think this is a reasonable way to define social interactions because individuals will reorient towards each other with only a few body lengths. And we've seen males turn and attack other males, or turn and then start courting, run up and court another female. So we think there's social information being passed within just a few centimeters of each other. So in total, with these scan samples, we observed over 1,800 social interactions for 250 males across these 9 populations. So this is our typical population, a subpopulation within the larger forest. All of these populations that we're looking at are within a kilometer of each other. Some are only a few meters apart. And this was all done in an old growth forest in southwestern Virginia near Mountain Lake Biological Station. So this is your typical subpopulation. You've got a couple of mating arenas, a couple of these brackets. Typically, our populations consist of about 10 to 30 males in a population. The sex ratio is 50-50, slight female bias. But we do have a couple of mega populations in this forest as well. So this log is just a small picture of the log that I could capture with my phone. My undergraduates have termed this the mothership. You can see all of these brackets along. There are hundreds of brackets on this population, and we have upwards of 160 beetles on this one population. But we wanted to include a fair sample of what we're finding out there in the woods, so we included them in our analysis. I looked at only male-male interactions for this paper. I'm sure there are lots of other interesting things going on with females, and we're processing those data right now. But we're going to look at male interactions here. And so this is our mothership network you can see here, and this is our other mega population. They form really well, tightly connected networks with a single module. But some of our other typical populations, you can see the network is broken into several different modules. And there are not a lot of repeated interactions. You can see that by the darkness of the line. There's only kind of 1 or 2 interactions between these individuals. So to look at differences in selection, we standardize all of the network metrics across these populations, so mean 0, variance of 1. And what this does is we're really looking at the most connected individuals in the population and comparing their fitness to the least connected individuals. We conducted generalized linear models for each metric, and we did this separately for strength and betweenness. So we're using a Poisson distribution. We check to make sure that our data were not over-dispersed. And we're using for a dependent variable here, our fitness component is the number of guards or successful inseminations. You can think of this as mating success. And for our independent variables, we included these things that we've seen to be under selection before. As well as the most important thing I want you to focus on here is the network metric by population interaction. So we're looking for differences in slope for those selection gradients. So here are our results. The things that we've seen under selection before were under selection in our meta population, but again, I'm pointing you here towards this strength by population interaction, saying that we're seeing differences in slope among the population. So if we visualize selection in these populations, you can see typically we're seeing kind of weak positive selection for network position. So individuals that are well connected are having high predicted mating success. But in our 2 mega populations, you see that we've got strong negative selection in those 2 populations. So we thought, well, what could be the factors that are causing these 2 different populations to have this negative selection? Forked fungus beetles are kind of a classic density-dependent model for sexual selection when we're talking about male body size. So we thought, oh, density might be really what's driving this. But if we plot each of the kind of standard linear selection gradients, so these are kind of Landy-Arnold selection gradients on the y-axis and density on the x here, we're not really seeing any pattern. So then we thought, okay, we better put in a bunch of different demographic variables into a single model. So we included density, sex ratio, and the population size into a multiple regression, kind of, as I said, a rough cut here, just to see if any of these predict it. And what we're seeing here is that sex ratio has kind of a loose explanatory power here on the selection gradient, but really it's population size that seems to be driving this effect. And by that I mean that our 2 mega populations have really different selection gradients than everybody else. So we can kind of split the two large populations and the small populations just to kind of visualize selection here. And the nice thing about this is we've got roughly equal sample sizes when we pull those two populations out, about 120 in each one of these analyses. And so you can see that in a large population, if you're a really well connected male, you've got low expected predicted mating success. But in a small population, if you're doing behaviors that make you really well connected, you've got high predicted mating success. So if we look at betweenness, this is that other measure of centrality. We're seeing essentially the same result here. We've got weak positive selection in a typical population, and in our mega populations, we've got strong negative selection. And we run that same multiple regression. We're finding that sex ratio and the number of males in a population is really what seems to be predicting these differences in selection. And again, we can split this up and visualize it. We're seeing the same patterns as we saw for strength here, maybe with a slightly weaker signal in the smaller populations, but still significant. So like a lot of science, I'm left with many more questions than I have answers. And so what I'd really like to do next is to see if this population size and sex ratio, if these are really the causal factors involved in these differences in selection. But I'm more interested in understanding the biology and behavior that underlies these patterns that I'm showing. So what I'm doing with this question is soliciting your just so stories so that I can go out and test them. I'll give you my favorite one, is that I think that maybe in the larger populations, these really well-connected males are interacting with lots of other males again and again, and they're having more aggressive interactions, and so they may not have the time or the energy to court and effectively mate with the females if you're really well-connected, whereas the ones on the periphery don't. But in the larger populations, I think that if you're well-connected, you're probably just bumping into a lot more beetles, and you can see how rare it was to actually interact with another beetle, and so you're interacting with more males, more females, you're getting more information and mating. But again, that's just kind of a hand-wavy hypothesis, let's put it that way. And so I'll leave you with the take-home message of this talk, is that the sign of selection on network position depends on the social context. And I find this really surprising because some of these populations are only a few meters apart. And so a male that disperses and lands in one population is going and behaves in a way that makes him well connected. He's going to have different predicted mating success depending on just which population he lands in. And I find that pretty interesting and exciting. And with that, I'll thank my funding sources and the large number of undergrads who helped me collect this data, and I'll take any questions.